Welcome. Today's video it is regarding the measurement and the quality check of the isolated DNA. The DNA it can be isolated from the bacteria, it can be isolated from the plant cell, animal cells, or the fungi or the viruses. Okay, when we are isolating the DNA from the bacteria, it can be the plasmid DNA or it can be the genomic DNA. So whatever the DNA it has been isolated, its concentration we have to find out. Okay, so to find out the concentration of the DNA, we can use the spectroscopy, and that it is a ultraviolet absorbance spectroscopy, or you can also use the UV visible spectrophotometer. Okay, so here the when we are measuring the absorbance in the UV radiation, we have to measure at the 260 nanometer. Okay, so the nucleic acids, it is going to give the absorbance at the 260 nanometer. And when we are measuring the absorbance at 260 nanometer, and if we are getting the absorbance 1, that is, if we are getting the OD as a 1, A260 of 1, we are getting, that is a absorbance measured at the 260 nanometer. It is a 1 OD, that is a 1 optical density. Then we can say that, okay, we can say that the double stranded DNA which is present into a given sample, it is of about the 50 microgram per ml. And if it is a single stranded DNA or it is a RNA, then its concentration, it is a 40 microgram per ml. Okay, so here for the measurement, okay, for the measurement of the concentration of the DNA, we have to go for the UV spectrophotometry, and in that we have to select the wavelength, and that wavelength it is a 260 nanometer. And when we are getting the 1 OD at a 260 nanometer, then okay, then the concentration of the double stranded DNA, it is how much? It is 50 microgram per ml, and the concentration of the single stranded DNA, it is 40 microgram per ml, and the concentration of the RNA, it will be the 40 microgram per ml. Now, here, this it is a standard one, and now from the isolated DNA, we have to find out the concentration. So you are taking, okay, you are taking the absorbance at the 260 nanometer, and if it is a 0.5, okay, the absorbance it is a 0.5, that is the OD, it is a 0.5 OD, then how you are going to calculate? Just you have to do the cross multiplication. 1 OD is equal to 50 microgram per ml, then how much for the 0.5 OD? So here it will be the 25 microgram per ml. As the OD, it is a half one. Okay, so in this way we can measure the concentration of the DNA by having or by measuring the absorbance at 260 nanometer. Now, next we have to go for the purity check of the DNA. So when we are isolating the DNA, that DNA, it can be contaminated with the proteins or the organic solvents like the phenol. So we have to find out that the DNA, it is a pure one, that is, it is not with the proteins and it is not with the phenols. Okay, and for that purpose, again here there is a use of the ultraviolet absorbance, that is a UV visible spectroscopy it is used, and the absorbance in the UV range it is used. And to find out the purity of the DNA, there is the measurement of the or the sample, it is measured in the range, okay, and that it is a 260 nanometer and a 280 nanometer. Now we know that at a 260 nanometer, the nucleic acid, it is going to give you the absorbance. And here in the 280 nanometer, okay, in the range 280 nanometer, UV range 280 nanometer, the proteins, it is going to give you the absorbance. The aromatic amino acids, it is responsible for giving the absorbance at a 280 nanometer. Okay, so by taking the ratio of absorbance at 260 nanometer by the absorbance at 280 nanometer, we can find out the purity of the DNA. And here, if we are getting, okay, if we are getting the ratio 1.8, Okay, if we are getting the ratio 1.8, then the DNA prepared, okay, it is a pure one, 1.8 to 2. If we are getting, okay, if we are getting the ratio of the absorbance at 260 nanometer by the absorbance at 280 nanometer, it is in between 1.8 to 2. Then the DNA, it is a pure one. But if, okay, but the ratio, it is a less than 1.8. If ratio it is a less than 1.8, then the sample preparation, it is contaminated with the proteins or the phenols. Okay, in this way we can check the purity of the DNA just by checking the but just by measuring the absorbance at a 260 nanometer and a 280 nanometer, and by taking the ratio of it. Okay, I think you may have cleared the idea about the measurement and the quality check of the isolated DNA. If you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.